Okay, remember, very important. In section B, you're going to answer 2 questions. 2. That means you're going to choose, say, 5 and 7. So I'm going to do those. And if you do 5 and 7, that means you got to answer all of these for 5 and 7. So 2 questions. Usually there's 3 per question. How many questions are you really answering? There's really 6 you are answering. So that gets confusing. Some students, they kind of want to believe they can choose, I'll do this A, I like that one, and then I'll do this B here. Okay, that's not correct. These are the questions, 5, 6, or 7, and whichever one you choose, you got to answer all of these. So all are required. Got to answer all of them. All right, so say you chose to answer number 5. Let's get into that. The first question is about explain how chemical energy for use in the cell is generated by the electron transport chain in chemiosmosis. So that's, if you see, that's 8 points. So that's going to be a long question. Okay, so remember, you can do an outline. This is kind of narrative form here, but you can also kind of do an outline if that's what you think is will work best for you. But you'll see I have that Na and FAD are reduced in the Krebs cycle to gain 1 or 2 hydrogen ions, basically making NADH and FADH2. That goes to the electron transport chain then. This is located on the inner membrane of the mitochondria. I went back and added that that's happening in the matrix. And then the electrons from the NADH and FADH2 are released to the electron carriers along that electron transport chain. And as the electrons travel along the electron transport chain, hydrogen ions are moved from the matrix to the inner membrane. I'm going to clarify that and say, probably better say pumped. From the matrix to the inner membrane space, and then we know these hydrogen ions become highly concentrated in the inner membrane space and eventually move back through the inner membrane via ATP synthase. And this is the chemiosmosis. That's called chemiosmosis. And as they do that, as the hydrogen moves into the matrix, it joins, I kind of mess up on that, it joins an oxygen molecule, and that makes water. And that's the reason you need to breathe oxygen. You need the oxygen for your electron transport chain. And that movement also releases energy and allows a phosphate to be added to the ADP, therefore making your ATP. Okay, this is energy for cellular use. For cellular use. So that's a pretty long answer. Again, 8 points. It's not going to be a short little thing. All right. The next one, 4 different functions of membrane proteins. Now you'll notice I put a list of 7. That's just to give reviewing, practicing. Don't put a list of 7. Just put a list of 4. They're only going to count the first 4 answers you give. So we see binding sites for hormones, cell-to-cell communication, channels for passive transport, pumps for active transport. Remember the sodium-potassium pump, the most famous. Cell adhesion, sticking to each other, immobilized enzymes. So the enzyme is just stuck on the inside of that cell. and then electron transport for ATP production, right? We've just talked about. So you'll notice that's 4 points. So basically you're going to get a point for every of the 4 different functions you correctly describe. Now remember, you can also get points for quality. So if you don't get, say if you only have 6 basic ideas here, remember you get points for quality. What that means, it's the examiner doesn't have to reread it. So no rereading. This happens to me all the time. When I'm grading your tests, I'm trying to figure out, whoa, what did they say? I can't quite understand it. And then some other students are like, wow, I get it right away, what they're trying to say. The quality, they don't have to reread it. There's no extra non-essential information in there. Some students will rewrite the question several ways or throw information from the question itself to try to, I don't know, just add more words to their response and hope the examiner will give them points for just writing words, but those words have got to be, high quality. Okay, so yeah, if you don't know all the answers or all the details, you can get points for the quality of the details that you know. Because that's an important skill. Oh, succinct. You know the word succinct. I forgot to see. So that's short and sweet. Short to the point. If you can do that, but explain it well, you might get an extra point for quality. It's only 1 extra point. The next question for the next sub-question for question 5. What is the difference between these 3 terms, anabolism, catabolism, and metabolism? And we see metabolism is all enzyme catalyzing reactions, including anabolism and catabolism. It is the broad word there. Anabolism is a type of metabolism that builds, synthesizes molecules from monomers. So you get individual molecules, you're building them into larger molecules. And then catabolism is the opposite. You're breaking down large molecules into smaller molecules. All right, so say, I chose 5 and 7. I just did 5. Now I'm going to answer 7. So I answered 5 here. And one thing that I didn't do on mine is I didn't really leave too much space in between. You're probably going to have plenty of space to write your answer. Okay, see all these pages. So don't worry about trying to make it too compact. So I would leave more space in between. I kind of jammed them all in together. So now I'm going to answer question 7. Turn the page. So this question talks about Mendel and Darwin, but discuss the role of genes and chromosomes in determining individuals and shared features of members of one species. And see, I didn't have it on camera, but you see the question, and it's worth 7 points. So again, it's going to be a long, answer. So it talked about it wants genes and chromosomes, not genes or chromosomes. So you got to talk about both. So I have genes can undergo mutations, which creates variations. Genes can also come in many varieties called alleles, which creates more differences. Individuals of a population of a species may share the same alleles or have different alleles. Some alleles are dominant and cover up recessive alleles in the phenotype. Remember phenotype and genotype? Make sure you know the difference between those. However, recessive alleles can be expressed in the phenotype when an individual has 2 copies of the recessive allele. All members of a species tend to have very similar genes and alleles, and genes are inherited from parents and passed on to offspring. So that pretty much covers genes. Then on to chromosomes. They're the structures which the genes reside at various locations called loci along the chromosomes. Members of the same species usually have the same number of chromosomes, like humans have 46, although not always. Down syndrome, people have Down syndrome have 47. Some people with, for example, Turner syndrome have 1X, so they have 45. Klinefelter syndrome have, I believe, an extra X, so they have 47. So they can vary. It's never 100%. Everybody has 46, but most people have 46. And I say that those extra chromosomes can arise due to non-disjunction, causing, for example, trisomy 21. That's a fancy way of saying Down syndrome. Sometimes gametes can receive an extra X or Y, or one fewer Y chromosomes. The sex is usually determined by the presence of a Y chromosome. Meiosis gives rise to a lot of new combinations of genes due to crossing over and independent assortment. So that's a lot of words. Remember, you're going to need a lot for those 7 points. So the next question is outline the process of speciation. So speciation, the splitting of one species into two, occurs because of some kind of reproductive isolation. And then reproductive isolation can arise from things like geographic barriers, differences in the time that mating occurs, that's temporal, and differences in the behavior, the mating traditions, the mating habits of the organisms under examination. And the gene pools become separated since the genes are not being shared, in the process of reproduction, and then they can become different. And maybe you could add in as mutations accumulate. So good to have some space here. I should have more space in between these questions. And these changes may take place slowly over time, which is called gradualism, or it might happen quickly, and that's called punctuated equilibrium. So it'd be good to add that in there. And then the last question is about homologous structures. Discuss one example, just one, don't do two, and just hope they pick the good one, of how they provide evidence for evolution. So homologous structures, they have similar structure but different function. They suggest they have a common ancestor, and the changes occurred through this process called adaptive radiation. An example here would be a human arm and a bat wing. The major bones are all there for both organisms: the radius, the humerus, the ulna, the carpals, metacarpals, and phalanges. They're all present. But they have different functions. Like we grasp tools. I can grasp this pencil. I can write with it. But a bat can't do that. A bat, it can only fly with its hands or its phalanges. Which is pretty sweet, Maybe that's better than writing. I know I would like being able to fly. That would be I think the sweetest superpower, in my opinion. That would be just to go with the clouds and stuff. Since they're so similar in structure, that's where that evidence for the evolution. See, the most likely explanation for the similarity in structure is that they were closely related at one point. Similarities. That is the most reasonable explanation. They share that common ancestor. So, you answer your 2 questions, you're done. So if you did 5 and 7, you don't have to do 6. But let me go ahead and I'll do 6. So those of you that chose 6 and 7, or what's the other combination? 5 and 7, 6 and 7, or 5 and 6, you can get that. Alright, so if you chose to answer number 6 as one of your 2, let's go through those. So the first thing, defense, your body defending itself, occurs on many levels. First one is describing immunoglobulins, which are antibodies. I'm not sure, it may be complicated to use immunoglobulins instead of antibodies, but I just stated they are antibodies, which are specialized proteins that are specific for the antigens on a pathogen. Remember, the antigens are just any chemical that are there existing on the cell wall or the coating of whatever kind of pathogen you're looking at. And these antibodies help to destroy pathogens by immobilizing that pathogen and attracting macrophages that help to engulf it. They kind of mark it for destruction. You see how I left space? I left some margins on each side so I can go back and add some things. So I think you should do the same too. So you can reread it and add some information. They're not going to freak out, the graders. Like you see, I crossed this out. Don't worry about that. Just get your information out there as best you can. It's not an exercise in the best writing necessarily. Remember, you can get points for quality, but that's not really what it's about. You can have terrible writing, not handwriting. They need to be able to understand what you're saying. But you know, they're looking for the information. They want to know how much you know about biology. So adding a little comment like this shows you know a little bit about biology. So don't be afraid to do that. Attracting macrophages to engulf the pathogens. They can also help to destroy the pathogen by bursting the cells of the pathogen. So remember, that was 3 points, I believe. So it doesn't have to be too long. The next question, outline how antibiotics offer protection from certain forms of infectious diseases. That's 4. Now, certain forms, remember, they always want to know that that antibiotics work on bacteria, not viruses. So you always want to, add that if you have a question about antibiotics, probably. So they kill or inhibit the growth of bacteria. Said that right away. The vital cellular processes of the bacteria are blocked, but not the vital processes of the eukaryotic cell, which is us, which I then said humans. So be more specific. Antibiotics disrupt the following processes of bacteria, aka prokaryotes: the metabolic pathways, DNA replication, transcription, translation, ribosome function, cell wall formation. Antibiotics do not protect against viruses, as viruses have none of these processes. Remember, they live off host cells. And some bacteria have genes that are resistant to some antibiotics. And the resistant genes can be passed on to new bacteria through reproduction and sharing of genes between 2 bacteria. So they basically make copies of each other asexually, passing that on, or they can form these little tubes which they can kind of do a form of sexual reproduction. They can send genes along to other bacteria. So that's B. And then for this one, the last one is the biggest question. Coughing. Some of you, my kids, are coughing right now. Oh, not COVID. They got some other nasty thing. But it says explain muscle contraction. So remember, muscle contraction. Oh yeah, that's a lot of big cycle. It's worth 8 points. So remember, you can, when you're explaining something like muscle contraction, it's kind of visual, put a diagram there, okay, and label it. These actin, thin filaments, you can get points for this. Myosin is the thick filaments. Maybe you'll even say this area shows up light on an electron micrograph. Shows up as a light band. Using a microscope, say. And this shows up as a dark band. So I'm getting information down. Okay, the myosin heads I labeled. And this would probably be relaxed. In relaxed state. Maybe you want to draw it in the contracted state. So you see my brackets are much closer together now, right? So this is contracted. So that's the contracted state. So that's good to have. So basically explaining the process, the muscle contracts when a nerve impulse arrives at the motor neuron on a muscle cell. We'll say sarcomere. And the nerve signal releases calcium ions in the sarcomere, which binds to the troponin, which then causes tropomyosin to move, exposing the binding sites of the actin filaments. Then the myosin heads form a bond, which is called, a fancy term for that is cross bridge. Alright, so sorry I take a break. Where was I? Power stroke. Okay, I think I had the that binding site being exposed because we had the calcium come in from the nerve impulse. The myosin heads then going to form a cross bridge. And then, on that myosin head, you have ADP, and that is released from the myosin, and that causes it to pull. That's called the power stroke. So it's the release of ADP from myosin head. That's really the key. That's where you have the movement. Power stroke, the movement. Okay, and then it's going to keep attached until an ATP molecule attaches to the myosin head, and that releases it. And then the ATP is then converted to ADP as a phosphate is released, from the ATP, and that causes the myosin head to go back into that energy storing position. As I like it, don't want to use a gun analogy, but a gun that's cocked back, and you create a hammer. Then it's storing energy. Okay, or like when you pull a bow and you pull it back, like that's a better one. So pulling a bow back. If there's still calcium there's still that nerve impulse, then it's going to bind and bind over and over again until the calcium leaves and the tropomyosin covers up those binding sites. Okay, and this contraction happens in the intercostal muscles, the abdominal muscles, and your diaphragm. So that's how you answer those questions. Make sure, write as clearly as you can. You can do an outline form if you want. I would try to keep it kind of narrative style like this, and don't be afraid to use diagrams.